And they say, well, you lost dopamine neurons, and this therapy is going to put them back with stem cells, which is a real... This trial looks at two different adaptive algorithms. Specifically, the multi-unit spike rate in the STN, across the population of neurons, was increased before a more conservative...
Welcome to Stimulating Brains.
Dr. Todd Herrington is a neurologist and the director of the DBS program at Massachusetts General Hospital. He specializes in treating movement disorders including Parkinson's disease, tremor, and dystonia, with a particular focus on patients undergoing DBS. Todd's research centers on the effects of DBS on the motor, cognitive, and psychiatric aspects of movement disorders, and he has employed intraoperative and noninvasive neurophysiology and neuroimaging to deepen our understanding of the basal ganglia's role in health and disease, aiming to develop innovative neuromodulation approaches for both motor and non-motor symptoms. Todd was recently strongly involved in the adaptive DBS algorithm for personalized therapy in Parkinson's disease, so the ADAPT-PD trial, which was led by Dr. Helen Brontë-Stewart at Stanford. This multicenter prospective single-blind randomized crossover study evaluated the safety and effectiveness of adaptive deep brain stimulation in Parkinson's disease. The trial made use of a new neurostimulator which can sense local field potentials and then react to these signals in real time to adjust stimulation based on the patient's brain activity. We also briefly cover stem cell therapy, the mechanism of action of DBS, and learn about lots of other really interesting insights from Todd. So I hope you will enjoy this conversation as much as I did. Thanks so much for tuning into Stimulating Brains. So Todd, thank you so much for taking the time off your busy schedule to do this interview. It's a big honor. We've known each other for quite some time and hold you very dear, and I know how busy you are, so it's fantastic that you could take out the time of your busy schedule.
Thanks, Andy.
We will have introduced you more formally by now, so we can dive right in. And as you may know, I always ask an icebreaker question about hobbies. So what do you do when not involved in medicine or science?
Yeah, thank you for having me. I do know that. I am a fan of your podcast and of your work in general. Anticipating your first question. I have still young kids, and so a lot of my outside of work time is spent with them skiing in the winter and hiking in the other seasons. But I do have a couple of hobbies that one I picked up more during COVID was sailing dinghy sailboats, little single person sailboats, which is a bit of a sport, but more for me a sort of meditative hobby that connects me with the water and the ocean. And yeah, it was something I had done in grad school a bit, but really got into during COVID when spent some time in Long Island. My mother-in-law has a place and it's near the water and some beautiful bays there that you can sail in.
Oh, great. Was about to ask, it's not on the Charles or on a lake or so, but it's Long Island. Okay.
I learned on the Charles during grad school, initially at a course through MIT and then subsequently through the community boating there.
Really nice.
I also fly dual line stunt kites, which is probably not a hobby that most people have heard about, but they're kites that are steerable via a couple of control lines. And
I know those.
Yeah.
It's great. I've never done it, but sorry to interrupt, but fantastic.
Yeah, it's another hobby that, again, it kind of gets you outside, and it's there's a skill element to it, and you can kind of make the kite do tricks, but it's mostly sort of a meditative hobby, I think.
Super cool. So could you share a bit about your journey into the field of neuromodulation? What inspired you to pursue the intersection of neuroscience, but of course also neurology and engineering? Was there an aha moment or any turning point that brought you to DBS or so?
Yeah. Yeah, a few. It's always funny when you look back on your history and you can draw a straight line and kind of imagine that it was all preordained. But of course, there's these little inflections where you imagine, like, if this moment didn't happen, would I still be sitting here? As an undergrad, I was studying biochemistry. I was doing research in bacterial cell signaling. And then I started getting interested in synapses. And then I got fascinated by the nervous system. I was just reading a ton about that. And I was applying to MD, PhD programs and was already thinking I wanted to do neurology. But I remember very clearly this paper by Miguel Nicolelis at Duke. First author was Chapin. And hadn't been interested in systems neuroscience at all. But this paper, uh, using recordings in the motor thalamus and motor cortex, multi-unit recordings, they trained a model to predict when a rat pressed a lever to receive a drop of water. And once they had their predictive model based on the cortical and thalamic activity, they then turned over the delivery of water just to that model. And a subset of the rats stopped pressing the lever. They just went up to it and started thinking themselves water. And I remember thinking, like, they created telekinetic rats. It was like
Yeah.
this amazing revelation. And it had never occurred to me that we were anywhere near a point where you could read out an intention like that from brain activity directly. And it blew my mind. And so then I started, reading really deeply about that kind of neuroscience. And when I went to my PhD, joined the lab of John Assad, who was a primate neurophysiologist, and I studied visual attention with him. But he was a real dear mentor of mine, still is a dear mentor of mine, a very careful scientist, but also a great communicator of science. So he's someone that could speak to a lay audience or group of medical students or graduate students or professionals very close to his field, and always deliver a talk that was just at the right level for that audience without ever really dumbing it down. Very sophisticated thinker. Uh, and while I was in his lab, when I was joining, actually just as a rotation student, had never done this kind of invasive neurophysiology at all, had a ton to learn. And the person that was sort of assigned to teach me, and I was helping him do some recordings, was a young neurosurgeon, functional neurosurgeon in the group, who was doing a research fellowship and had just started to establish his lab at MGH. It was Ahmad Eskandar.
Oh, wow. Yeah.
And he taught me to do in non-human primates single unit recording. And then similarly invited me to come with him to see a case in the operating room, a DBS case that he was doing. And several things about that experience blew my mind. One was that the techniques they were using to do DBS microelectrode recording were virtually identical to what we were doing in the research lab.
Yep.
And actually in that project we're recording from the basal ganglia, so that the translational potential of both my PhD work that I was doing, as well as the potential for research applications, that intraoperative data were just really obvious and fascinating. Plus, I got to see a successful DBS, and I think many people who ultimately decided to pursue this career have had that moment when they saw the DBS be turned on and the tremor melt away and the hand start to open freely and the patient had this huge smile. And so I had that moment, and that planted a seed where, even though I was just at the very beginning of my PhD at that point, DBS was always front of mind from that on. And so here I am, a lot of years, of years later.
Leading the DBS program at MGH. That's really fantastic. I did not know that story. And you mentioned John Assad and also Ahmad Eskandar as, I would say, turning points or mentors. Were there other people that you should mention or you want to mention on your path that helped?
So many. Those two, I think, on my scientific journey into DBS specifically, were really critical mentors. I've, of course, had many clinical mentors through neurology residency, and and fellowship. I think the director of the Movement Disorders Fellowship at MGH is Alice Flaherty, who some might know through her writing, and she has a real focus on the intersection of neurology and psychiatry, and working and previously directed the neurology half of the DBS program before she ultimately ceded that to me. But I've learned just immensely from her about in my mind sort of breaking down the barriers between treating just the movement disorder symptoms versus the broad array of psychiatric symptoms that come along with movement disorders, as well as just thinking openly about the potential psychiatric impacts of DBS, even in movement disorders patients, which are sometimes quite difficult to nail down, but much more so now than even 10 years ago, well recognized.
Yeah, fantastic. And then I remember in your in your time while Ahmad was also still at MGH, you did really cool things on decision making and on stimulation and invasive, not invasive, during a task, interfering with DBS. Do you want to talk a bit about that phase of your career scientifically?
Yeah, so very collaboration with Sean Patel, who was a graduate student and then a postdoc with Ahmad Eskandar, had done using these multi-unit microelectrode recordings in the operating room in a pretty simple gambling task, like a card task, kind of like the child's card game War. High card wins, and you had to make a bet as to whether you think you had a winning hand or a losing hand. And what was striking about it is that in the STN, there was encoding of the decision to make a higher risk or a lower risk bet, but specifically that encoding was present in the multi-unit spike rate when the individual had a high conflict scenario. So it didn't encode that decision in all scenarios, but just specifically when the right answer in terms of whether you should in this situation make a high or low bet, when that answer was not clear. And specifically the multi-unit spike rate in the STN across the population of neurons was increased before more conservative bets. And reason that, that's interesting, and maybe that is something that could be replicated if you could deliver a burst of high frequency DBS during that decision period.
Yeah.
So used people then that had Medtronic, was the only company at the time, implanted DBS systems and jerry-rigged a system where we could use a computer to quickly turn the DBS on and off just for a defined period of about a second right around that decision period, and actually showed in a cohort of people that would deliver that little burst of stimulation, particularly during those trials where the correct answer in terms of a high or low bet was not obvious, we biased them towards making a bit more conservative decisions, which was a super cool thing to see.
Yeah, totally cool. And it reminds, like when you mentioned the work in primates, it does have this, very scientific component where DBS really was used as a tool to study the brain more than maybe as a clinical treatment. But of course, you do direct the program, so you also do a lot of clinical applications of DBS, and we did talk about some of your colleagues. Sean Patel was one, and DBS always brings together very diverse fields. So, collaborations with engineering, neuroscience, and then also clinical practice. How did that generally influence your thinking of what to do with DBS in terms of science, but also, about the clinical work? So in other words, the crosstalk between, science and clinics, how much is that happening?
That's a fascinating question. I have found myself, in terms of how I spend my time, with a variety of different kinds of projects, some of which are more sort of exploratory and some things that have branched off of that that decision-making work, and we're doing some work in the EMU now that's trying to explore those circuits around decision-making more deeply. But a lot of my time is spent in doing things that are more directly adjacent to clinical care and sort of analyzing DBS-related data, be it imaging or neurophysiology, and try to think about how to integrate that into care. And I do feel like we're at this golden era right now, maybe of neuromodulation in general, where there's so many areas outside of DBS that are moving forward very quickly. But specifically within DBS, I think it's extraordinary that we have these 3 companies that are approved right now, several more that are approved around the world to deliver this therapy. But if we just look at from a US lens, 3 companies that are approved to do this, all of whom are quickly innovating and delivering new approaches.
Yeah.
So Abbott with telemedicine enabled remote programming, Boston Scientific with image guided programming, really integrating that into the tablet experience, and then developing, I know I'm sure near and dear to your heart, and in no small part based on your work and work you've enabled, enabling an automated suggested DBS program based on imaging locations, right? And then Medtronic with sensing enabled DBS, and now adaptive DBS. So it has brought me great joy to try to quickly adopt each of these new technologies and to experiment with them in clinic. And I feel like that's been one of my roles here, is to try to facilitate that, both my own practice and for the nurse practitioners that have worked and helped care for our DBS patients, as well as for other people as they come and learn about DBS, to try to lower the bar, the barrier to using those technologies in clinical practice as much as possible.
That makes a lot of sense, right? Even just staying on top of the game with all the new things that are coming is not a trivial task. And you have had, we'll get to that hands-on experience, also very much in the sensing world with Medtronic. Before we dive into that, though, you in 2016 you wrote a review or a perspective, or like a summary paper about the mechanism of deep brain stimulation. And that is always very fascinating, because I think there's one end of the spectrum says we just don't know how DBS works, and then there are others that simplify it and say it's a lesion, and then there's a lot in between, and it probably exceeds a podcast format to talk about this exhaustively. But if you have a few 2 cents to give on how does DBS work, that would be super interesting.
Yeah, it's amazing to think that paper is almost 10 years old now, and easily this could eat up the next hour. It's such a fascinating thing that we still don't have this nailed down. When we wrote that paper, we called it mechanisms, plural, very intentionally, because it's clear that while we don't understand all the ways in which DBS is working, that it's not working via a single mechanism. It's not even working, I think, even if you focus down on one target like the STN, it's not working via one structure, even in or around the STN. So I think your recent paper looking at individualized hotspots around the STN for different Parkinson symptoms highlights that we're modulating probably different subcircuits, different fiber pathways into and out of the STN, or passing by the STN to modulate different symptoms. I think no thinking about the mechanism of DBS can escape the fact that through first approximation, DBS looks a lot like a lesion.
Yeah.
Right. And so I still feel like the informational lesion hypothesis, the idea that there is a dysfunctional, probably in many cases related to the loss of dopamine in the sensory motor striatum, a dysfunctional circuitopathy that the basal ganglia are important in maintaining or perhaps even generate, and that you do better by taking a node of the basal ganglia largely offline than leaving it online and being dysfunctional. But I think also that as I've thought about this over the years and had experience programming DBS, I think almost certainly a lot of what we're stimulating, let's just take around the STN, a lot of what we're stimulating there is not helpful, right? Is disrupting normal functional activity in those regions and throughout the network. And so I think when you look at like cervical dystonia and palatal stimulation, or other focal dystonias, and you see that there's a not insignificant rate of induced Parkinsonism or fine motor control problems that almost certainly, to my thinking, are also happening in Parkinson's disease. But the positive impact of DBS on people with Parkinson's disease is so great that you don't appreciate that you're also interfering with these normal functions. I think speech is like that a lot of times. It can slide in there as a sometimes more subtle and sometimes quite dramatic side effect of stimulation that is overwhelmed or just becomes not the focus because the other effects of DBS are so striking. So for me, I'm looking at those literatures and hoping that over time, as we start to deliver this therapy in a more targeted way, we can take away some of those off-target effects. I also think that there's ways that we're using DBS now, just starting to use it, that are going to work in a fundamentally different way. So I'm really eagerly following the dentate nucleus stimulation literature, for example, around stroke rehab. Andre Machado's work.
And that's low frequency, right? That is likely very different in mechanism.
30 hertz, right? Totally different, right? And I think there's people who don't understand whether that's maybe, does it just kind of stimulate trophic mechanisms, or is it something like a stochastic resonance process where you're just taking a circuit that the gain has fallen on it owing to the lesion, but there's still important time varying activity in that circuit that could be functionally helpful if you could raise it up to a level where it can be heard above the noise. And so I think there's other ways of using it, of using neuromodulation that we just haven't begun to really tap in the real world. And I often go back to the work. I think you've had her on the podcast, Aaron Giddis's work in the pallidum in the GPE, where again in rodents, and they're working hard to try to replicate this now in humans, but circuit manipulations of specific subpopulations of neurons in the GPE to produce these dramatic, long-lasting improvements in Parkinsonian symptoms in the rodent, as well as Parkinsonian physiology in the rodent. It sort of harkens back to the coordinated reset neuromodulation results. Make me feel like fundamentally there are, and how long it takes us to really implement these in humans, much better ways of manipulating the circuit than just 130 hertz slam on the gas kind of stimulation. So I don't know, that's how I'm thinking about it these days. I'm curious how you think about it. Put you on the spot.
I mean, I think I very much agree with what you just said. I think I am probably one of these more simple-minded people that think of a lot of the DBS effects really resemble a lesion, but only if applied in that specific way, right? 130 hertz. And again, the focus should be on this informational lesion, because of course we do also elicit action potentials, but if they fire along with the 130 hertz pulse, the information content will still be reduced, or the bandwidth of that circuit will be reduced. So I think totally agree with you on first approximation, at least, with a lot of ifs and buts and add-ons. It, like the chronic stim is really lesion-like, but the cool thing is, and that will follow into exactly what you just said, is that we can turn this lesion on and off all the time, right? And we could do so in a very rapid alternating way. We'll get into closed loop, and so if you think of this as a brake steering a car down a hill, you can do a lot with just one pedal if you do it in a very elaborate way, right? If you maybe can react to things, and with a real lesion, of course, you can't. It's permanent, and so I very much agree with you that there could be this essentially second era of DBS coming with all these pulsed and burst-like simulations, but also adaptive simulations. So very much looking forward to it. I think we're not there yet for most things, but there is a lot of potential there. So, yeah.
I agree very much with that. The other thing that we is easy to overlook is the different time course by which symptoms respond to DBS, even the same symptom. So take something like tremor, which I used to think very much of, like tremor is a ongoing oscillation in the brain that we interrupt in real time with DBS. And that looks to be the case in for example, STN or VIM in most cases, or at least the clinical response is consistent with that. But then you move to the pallidum in Parkinson's disease, and tremor response can be very clearly delayed, right?
Yeah.
Michael Oken and the UF group have a nice series on this, and we certainly see it to the point now where if I don't see good tremor suppression doing an initial programming in the GPI, I don't even worry that much. You say, well, let's just do it based on bradykinesia and rigidity, and then we'll see what emerges. So clearly, that's a different mechanism by which tremor is being interrupted in those patients. Some other plastic change in the circuit, which makes it no longer supportive, conducive to this tremor oscillation, but not a direct suppression of it. So much yet to learn.
Yeah, I totally agree. It could still be lesion-like, though, right? With pallidotomies, potentially, it could still, like, a lesion can still produce plasticity as well, in a way.
Absolutely,
But yeah, much to learn, as you say. So before we go into the ADAPT trial, in 2020, you were also co-first author on a New England Journal of Medicine paper that implanted patient-derived dopaminergic progenitor cells, which had been differentiated in vitro from autologous-induced pluripotent stem cells, so iPSCs, in a single patient with Parkinson's disease. I think this was really a breakthrough of a feasibility trial, but also did show some really promising results. Could you summarize how that trial came to be, and then also what you found?
Yeah, absolutely. Yeah, I mean, this, the cell therapy field is in Parkinson's disease remains really controversial. There are people in the Parkinson's world who feel like this is a tried approach that doesn't address a clinical need, and then people that are extremely bullish about it. And it's really a busy field now. Maybe it's useful to think back through the history of it a little bit, and that gives both cause for skepticism and cause for hope. The simple idea that you're missing dopamine neurons in a part of the brain and you should just put them back, right? Surgically put them back. It's been around a long time. In like the 1980s, people were doing autologous adrenal transplants, putting in catecholamine neuroendocrine cells, and that effort failed. And then in the 90s, there were xenotransplantation approaches with fetal porcine midbrain transplants, and that was not successful. And then there were human fetal tissue transplants in the 90s that open label results looked really promising, including some dramatic long-standing responses, and 2 randomized controlled trials in the early 2000s that both failed to meet their primary endpoints. And that was the sort of end of the field for a while. That was never really going to be a practical approach anyway to treat Parkinson's disease using fetal tissue-derived cells. But in addition to the not meeting their primary outcomes in terms of UPDRS improvement, people have these graft-induced dyskinesias, which were involuntary dyskinesias that persisted even when levodopa was dramatically lowered or withdrawn. And since then, there's been a lot of work trying to understand, model what the origin of those. Obviously, the original idea was that, well, graft-induced dyskinesias are just excess dopamine that you can't control from these grafts. But there were a number of things that pointed to alternative explanations. The amount that your UPDRS improved did not correlate with developing graft-induced dyskinesia. You might expect that if you had a better response, you kind of overshoot. You have a really good response to bradykinesia and rigidity, but then you have dyskinesia, kind of like an excessive peak dose response. But that didn't seem to pan out in the data. There was not a correlation between the change in PET signal of dopamine integration, the fluorodopa PET, and the development of graft dyskinesia. So it didn't seem to correlate with just the amount of dopamine synthesis activity. And then there were serotonin receptor or the reuptake inhibitor or the presynaptic transporter. Yeah, serotonin transporter is what I'm going for. PET ligands that in very small number of the patients did seem to correlate with graft-induced dyskinesia. So people that had the graft-induced dyskinesias had higher concentrations of the serotonin transporter. And then even a serotonin agonist that acts on presynaptic serotonin receptors and so it's thought to decrease serotonergic signaling, buspirone seemed to suppress graft-induced dyskinesias. So all at the end of this field is like, well, we're not exactly sure. It doesn't seem to work that well. But maybe these graft-induced dyskinesias were related to inadvertently transplanting serotonin neurons. And you still had, coming out of that era, this subset of patients who had dramatic, long-standing, durable improvements in their motor symptoms. So then the big advance that's happened since then is the ability to more efficiently generate induced pluripotent stem cells from human tissue.
Yeah.
In the case of this particular study, fibroblasts, and to develop a more pure population of dopamine progenitor cells that you could transplant back into the brain. And so then a number of groups have moved forward to try to test this hypothesis. I got involved into it in this study really kind of midway through when it moved to Mass General. So it really begun with Kwang-Soo Kim, who's a stem cell biologist at McLean Hospital, and it developed a lot of the underlying technologies that were the stem cell technologies that were being assessed here. And then he had been collaborating with Jeffrey Schweitzer and Bob Carter in California, both one a functional neurosurgeon. Bob Carter was the head of neurosurgery at UCSD at the time. And they were well on their way and actually had even done the first hemisphere transplant when they brought this project with Bob Carter coming to Mass General as the chief of neurosurgery at Mass General, recruiting Jeff Schweitzer to come with him, and they brought this project and it finished up here. And you're right. It was interesting to see it at that stage. I counted myself as somewhat of a skeptic of the cell therapy space at that point. I think we have to be really careful interpreting anything that we see in that first study because this is an open label study of a single patient. But we observed improvement on the order of 20 or 25% in both the on-state and the off-state UPDRS. There were some issues there. They didn't have great baseline data, particularly in the off-state, so that undercut some of the conclusions there. And there were also trends towards improvement in the fluorodopa PET signaling that suggested these cells had survived, and those increases were more were localized to the posterior putamen where the transplants had occurred. But what I was struck by is there were a number of domains of just day-to-day function that the patient improved in, going from wearing Velcro shoes back to wearing laced shoes because he could tie them more easily, and resuming swimming again, which had been a favored activity. And so it certainly felt like things had moved in the right direction for him, at least over that period of time. But of course, really you need larger studies. And so, in the meantime, we have that effort has gone on. There's been continued advances in these technologies, and we actually have a IND from the FDA to start a phase 1 trial of this, which actually we're intending to start very soon, maybe as soon as next month.
Oh.
But the technology, I still think there are major technology development that will benefit this approach. For example, we've published all the preclinical data for that individual patient, and that led to the approval of the IND. And then a number of companies, Blue Rock Therapeutics is well into a clinical trial of allogeneic cell therapy. So this is using a singular cell line and implanting it into all patients, with the idea being there that go ahead.
Would that require immunosuppressants then? That
Right.
case? Yes.
The logic is you require immunosuppression, whereas the autologous approach, the logic is those cells we recognize as self, and so you would not require immunosuppression.
So in your patient, I remember from the paper, the patient did not require that, right? Because it was...
That's right.
Yeah. Okay.
No immunosuppression, and they... It appeared that the patient didn't develop an immune response to the cells based on a humanized mouse model.
Yeah.
We could talk through that experiment, but that was encouraging. Again, N of 1. There's another company, Aspen, that is moving forward with an autologous approach, so similar to what we're doing. But all these groups and other groups that have tried to do this in animal models struggle with the variability in terms of the fraction of dopamine neurons that you transplant that actually survive, which even in carefully controlled animal experiments varies considerably. So Kwang-Soo Kim had a Nature paper just in 2023 showing that some of that variability is caused by the early immune response to the surgery itself, which leads to a few funny things. One is that a lot of the cells you transplant actually turn into glia, which is strange given that these are dopamine progenitor cells that are in vitro, well on their way to becoming dopamine neurons. And then a lot of them die. And so you really, to get the number you want to survive, you implant many millions of cells to get 20 or 40,000 surviving at the end, dopamine neurons. And so by co-transplanting those cells, again, now this is an animal model, with T regulatory cells that were also autologously derived, you could markedly shrink the overall size of the graft, fewer cells turning into glia, and double or triple the number of dopamine neurons that were survived there. So, I think that these technologies are going to keep evolving, and we will see ultimately if this can be impactful enough to merit inclusion in the broader array of clinical treatments that we have. I think this would probably not be a popular take from the companies in terms of what they want, where they want the bar to be set. But from my perspective, if cell therapy works as well as DBS or as well as, say, subcutaneous levodopa, Vyalev, which it basically just controls off-state symptoms, basically acts like a source of, you can put a catheter with dopamine and you leak it into the posterior putamen and get the same result. I think ultimately we're going to struggle to justify the complexity of this therapy, given that other kinds of approaches to achieve that are going to continue to advance. But one of my hopes is that if we're truly able to replace the dopamine circuitry in a way that's functionally meaningful, and if you look at people that have DBS, and you often largely control their off-state symptoms, and they spend most of their time in something closer to their good on-medication state. And if you look out over, say, 5 to 10 years, and Patricia Limousine has a really nice series looking at this, their off-medication state remains pretty similar. Like DBS is still controlling that really well, but where they give up the most is in their on-medication performance. Right? You're less responsive to levodopa. You no longer achieve that same peak performance. And one, I think, reasonable model is that relates to the sort of terminal degeneration, where those last few remaining dopamine terminals in your sensory motor striatum are dying off. And if you could restore that, so restore someone's response to levodopa, restore their response to other neuromodulation, that would be a really meaningful improvement. And that requires demonstrating that you can improve on-state function with these therapies, which I don't think we have any therapies yet that actually do that. So that's my hope, but I think it's an open question as to whether that's possible with these therapies.
That's a great point, because my next thought would have been, so the dopaminergic neurons that die are in the nigra, not in the putamen, right? Mainly. And so we know that a single dopaminergic, like physiologic dopaminergic neuron projects to big portions, a single neuron to big portions of the striatum. And it seems like a very intricate way, although maybe not as specific, right? It's like one neuron will go unselectively to big parts of the striatum. And if you now put new dopaminergic cells into the putamen, then of course you have more dopamine there, but you don't have the inputs to these neurons, right? So it will probably never completely replace the circuitry, because for that you would need the inputs going to the nigra and then the projections to the putamen. But what you're saying is that if you don't have these projections, then the receptors and the receiving cells will also die off, or will not be, and then levodopa won't work as much anymore. So if you would just try to essentially keep the receiving cells in the striatum receptive to dopamine, that alone would be super helpful. That makes a lot of sense to me. I never thought of that like that way.
Yeah, but you highlighted what is the huge drawback to this approach, right?
Yeah.
And why it is so important also for people with Parkinson's who are thinking about these kind of therapies, to understand that you're not truly restoring the original circuitry.
Yeah.
Right? You're making something different that we hope could be better. Absolutely. Yeah. And there's lots of important, there's a whole, dopamine is perhaps the most studied neurotransmitter in the brain. There's so much intricacy into the way dopamine is released, and so many important functions that are coded there. And, without a doubt, there's no way this therapy could replicate all of those functional aspects of dopamine. I also see this technology as a platform technology where, dopamine, because of what you mentioned, the relative simplicity of the dopamine circuit, that it's not like you're trying to replace a particular population of neurons in a cortical column. You're replacing something that already sends really broad projections.
Yes.
It's maybe the lowest hanging fruit for being able to restore some part of a circuit.
That's a good point.
But there's no reason that we should be limited to just trying to replace dopamine neurons and just act like dopamine neurons, right? Why not have them do other things? Like, you go back to Aaron Giddis's work. Could we not put in a cell population that could replicate these parvalbumin versus limb-homeobox 6 dissociation in the physiology, right? Integrate with the circuit and push it to operate in a more functional way. And, there's a lot of steps between that idea and actually something that you can put into humans. But I think just the idea of figuring out how to get cells manufactured efficiently in the lab and into people in a way that they survive, there's a lot of value there that we've only begun to try to unlock that.
Super helpful conversation for me to get a good overview, a glimpse of an overview here. Last question on this, and you might not know, but since it's published in 2020, do you know how the patient is doing? Is there any? So West Coast or East Coast?
West Coast.
Okay.
Still in close contact. I feel like he's a known individual. Like, he's been public with his experience with this. I want to be a little bit deferential to that and not say too much outside of what we've agreed to publish. Except maybe just to say that I think that the impact was durable, but I think that everybody would agree that it hasn't stopped the progression of the disease, right? And so I think that's another. I don't know if how often patients listen to this podcast, but when I'm talking to patient groups about this kind of work, I think it's really important that people understand this is not a cure for Parkinson's. And I think this comes up a lot because people generally very strictly associate loss of dopamine with Parkinson's, when of course there's much more going on. And they say, well, you lost dopamine neurons, and this therapy is going to put them back with stem cells, which is a real magic word, right? That grabs attention, and they think that's a cure, right? What could be more of a cure? And it's quite clear that that our goal for impact is, more specifically motor function, very specific functions. There's a lot more that's happening in Parkinson's. So, yeah.
Okay, moving back to DBS. This was a bit of a detour, but super interesting. Thanks for sharing. And we thought about recording this session mainly with the focus of the ADAPT-PD trial, which is probably the most exciting thing that has happened in the entire field of DBS in the last years probably, and is now ready for prime time. And maybe for those new to the field, could you explain the core concept behind the ADAPT-PD trial, even maybe also what adaptive DBS is? I think most people will know, but maybe a brief intro would still help.
Sure. Yeah. So adaptive DBS, as opposed to what we call continuous DBS or CDBS, which is the typical way DBS is programmed, is that you set a given stimulation configuration of contacts, anodes and cathodes, and a given pulse width and current and frequency. And then your stimulation just stays at that level continuously, usually 24/7. Sometimes people make little adjustments to it. But we recognize that people with Parkinson's in particular are not in the same state at all times. There are times when their medicines are peaking or when their medicines are falling. They're sleeping, they're waking, they're active, or they're resting on the couch and watching a movie. And the need for DBS, like the level of current, let's say, that you require in these different states, is likely to be different.
Yeah.
And from a programmer perspective, I think we often calibrate the DBS, or we push the stimulation up until we have addressed all of the off symptoms, or as much of the off symptoms as we can, which are often the most bothersome symptoms that people experience. And we try to keep it below a threshold where it's causing overt side effects. But there's this general idea that maybe we're causing more side effects even than we realize, at least when they're subtle. And so the idea of adaptive DBS would be, do you have some mechanism to titrate how much DBS therapy you're delivering at any given moment in time to be just enough, just the right amount and not more? And in this trial, it was focused on one particular neurophysiologic biomarker, oscillations in the alpha and beta band, which I imagine the listenership is very familiar with the beta band oscillations and the fact that they correlate with bradykinesia and rigidity severity, that the degree of response of beta band oscillations in terms of power reduction correlates with how much you respond to levodopa therapy and to DBS. And so the logic was, could you use the power in that beta band, an individual patient's beta band, to titrate how much DBS therapy you deliver in real time? And I mean, this idea and developed gradually over really decades, and there's a ton of people that contributed to that. Simon Little and Peter Brown, one of the earliest papers really demonstrating this with the particular approach, that a version of that was included in the trial called single threshold DBS. That was way back in 2013, which I think was the same year that the Medtronic PC plus S device was released, and so allowed it to start doing more ambulatory recordings and develop the data set that ultimately led to the design of this trial. But basically, this trial looks at two different adaptive algorithms, two ways of trying to map the beta power onto your DBS therapy. And one that was called single threshold DBS, because there's only one threshold parameter that you set, is based on the idea that beta power occurs in bursts. And so there are periods of relatively low beta and then periods where the beta spikes. I think, it's interesting. I mean, that is very clearly true, I think, in human and non-human primate motor cortex in the healthy state. But in the Parkinson state, maybe it's a little bit more variable, but certainly beta is highly variable on sub-second timescales. And so the question is, could you just, when the beta is at the high end of a range you set, jump up to a higher level of stim quickly? And when the beta is below the threshold you set, could you back off quickly? And the idea would be that you could adapt multiple times per second to your high level of stim, your low level of stim, but you don't really arc anywhere in between those. You just have two levels that you are alternating between. And so if you think about on a longer time scale, hours, your quote high or low stimulation would really be about your duty cycle, be how much of the time, what fraction of time do you spend at your higher stim level versus your lower stim level.
Yeah.
Right. And then the other approach that was called dual threshold, because you set two different thresholds that define a range of beta power that you have decided is acceptable or optimal.
Okay.
And if the beta power is in that range, then you do nothing. You leave your stimulation where it is.
Yeah.
If the beta power climbs above that range, you increase stimulation to try to suppress it back into the acceptable range. If the beta power falls below that range, then you back off on stimulation by decreasing stimulation current until the beta power comes back up in the range that you've set. And each of these methods involves obviously setting an upper and a lower stimulation limit. The algorithm will not drive it beyond those limits that you set based on clinical criteria.
And would the second one then essentially have three different stim settings, right? Or like would you predetermine of the high amplitude, the mid, the normal range, and then the low amplitude setting, or?
So this particular algorithm, you set two stimulation levels.
Okay.
The lower stimulation limit, which is the lowest stimulation that the system will ever deliver.
Yeah.
The upper stimulation limit, which is the highest that can be delivered. And then depending on how far apart, let's say those are a milliamp apart, in 0.1 milliamp increments, the system will go up or down until it either drives the beta band power, the control signal, into your acceptable range, or it hits one of its limits.
Great. Okay. Interesting.
So it really can live anywhere. So there on hour to hour, if you think about how this thing changes over hour to hour, people say as people come on or off of medicine, as opposed to this duty cycle idea for single threshold, it really is the absolute level of stimulation. So if you're going between 1 and 2 milliamps of stim, maybe when you're on medication and you're at your peak dose and you perhaps need less stimulation, it's hanging out near 1 milliamp. And then when your medicines start to wear off, you need a little more stimulation, the beta power increases, the stimulation creeps up closer to the 2 milliamp.
Okay. Understood. And how was the design of the trial? Did everybody try both versions and also maybe the continuous, or how did you select that? Yeah.
Yeah. Well, so I had no hand in the trial design. So
you guys, yeah.
Yeah. I got it. Yeah. So this is this was a multi-center trial. There were 10 centers across 4 countries.
Yeah.
Helen Brontë-Stewart at Stanford is the global PI of the study, and the design of the study is that we took people that were already programmed and stable on continuous DBS.
Yeah.
They came in and they went through a baseline assessment where you had them do a 72-hour symptom diary and the usual sorts of Parkinson's surveys to capture their function on continuous DBS. And then we tried to program both adaptive modes.
Yeah.
So for every patient, you tried to program both as best you could, both the single threshold and the dual threshold. And if both modes could be successfully programmed, by which you find that the participant and the investigator felt like the mode was functioning adequately. If both modes were successful, then they were randomized in a single blind fashion to single then dual or dual then single.
Mm-hmm.
If only one mode was successful, then you just move forward with that mode only. And if no modes were successful, then you dropped out of the study.
Okay.
And then to assess the modes, you had a month on that mode. And then you, again, symptom diary outcomes and the usual questionnaires. And then at the end of that, if the patients could choose to either continue on adaptive DBS, or if they had tried both modes, they could choose which one they felt like was more effective for them.
Yeah.
And they continued that for an additional 10 or so months of long-term follow-up. And at the end of that, they can continue into what's called extended access, where because these settings were not available on commercial devices in the U.S. and Europe or Canada, they had to be in the study to continue on adaptive DBS. But as Papa will talk about, most patients remained on it through into that extended access phase. So the benefit of this design is that it really captured a real world population of people that are already being treated with continuous DBS. It's really a diverse population with about 2/3 STN and 1/3 GPI. It involves both the newer Medtronic sort of sensing optimized leads called Sensite, as well as the older 3387, 3389 electrodes. And it's a mix, right?
Yeah.
The main, I think, drawback to this design was that the comparison between CDBS and the ADBS modes was not blinded.
Yeah.
Right. So participants knew that they were on an adaptive mode, and so you have to take that into account when you think about all the assessments.
Interesting, great. And then what did you find? So lots of questions, of course. Which mode was maybe better? How many patients stayed on adaptive mode?
Yeah. So at this point, these results were presented initially at annual meetings in the MDS in 2023, and then MDS again in 24, and AAN. So, and hopefully the paper is coming out very soon. But what was presented at those meetings, looked, in my opinion, pretty good. What I'd say is, it's 68 people enrolled in the study overall. 52 went on to get some ADBS programming. And so people dropped out initially for a variety of reasons, mostly because they don't have the right signals that are adequate to power ADBS, or you have artifacts like ECG artifact that interfere with your ability to use the signal. So I think one important thing is just you think about, is every patient with Parkinson's going to be a candidate for this? There are some requirements in terms of having the right signals, but also making sure that your optimal stimulation contact has sensing enabled contacts with the right signals around it, right? So people may not be a candidate for this approach because of those constraints. Ultimately, 45 people could be programmed in at least one of the adaptive modes, and 30 could be programmed in both. So you had head-to-head comparison for 30 people.
Yeah.
So there was a total, in terms of like who completed evaluation months, there were 40 people that were evaluated in dual threshold mode, 35 in single threshold mode. And if you take as your metric of what was more successful in the long run, there's about a 60-40 split in terms of dual threshold being selected by patients more frequently than single threshold. But a good number, a good fraction of patients chose both, right? So single threshold was a little less popular, but was 40% of the participants. The primary outcome measure for the study, so the outcome measure that was set with the regulatory bodies, was a bit of a weird one. It was a performance metric where the goal was that at least 50% of the participants would find that should have ADBS performance that was similar or better than their CDBS performance.
Yep.
So what does it mean to be similar or better for a single subject? The idea was to use the diary, the 72-hour symptom diary, Hauser diary, to record. I think probably all your listeners are aware of, but you basically rate in half-hour increments whether you're on, you have troublesome dyskinesias and on, whether you're off, whether you're asleep. And then you have this problem where, what does it mean, given the variability in that metric, if you have an hour more or hour less of on time, is that meaningful? And so they applied this criteria where they looked at the difference between ADBS and CDBS across the whole study. They just looked at that distribution, and they defined as one standard deviation based on that distribution. And then they said, okay, if you're within one standard deviation of your CDBS performance or better, then you're considered, performing well enough. And over 90% of both people that tried both modes performed at that level based on the diary criteria. The other metric, you say, well, what about just the raw change in on time, right, in terms of hours? And if you look at that, and you look at the mode that people selected to stay in long term, they gained overall about an hour and a half of on time per day on ADBS.
Yeah.
So on one hand, it's impressive because these are people that are already optimized on CDBS and we're doing pretty well at baseline.
Yeah.
On the other hand, we have to be a little bit careful because this is an open label comparison,
sure.
right? And maybe perhaps the most telling thing is that over the long term, 44 of the 45 people that entered the ADBS evaluation period decided to stay on it for the long term.
Yeah.
So it's, patients embraced it and continued on it for a long-term follow-up, which was positive.
That might really mean a lot, right? I think there's even voices sometimes where I think there's one investigator in Munich that told me, Kögelsberger is the name, that he, asks patient what feels best, right? Which setting feels best, and that might even be a programming strategy. And I think never investigated in a real trial yet, but there are plans for this where the idea would be, maybe the interoceptive component of a patient is as good, maybe, or similarly good as clinical scores. Does that match your general feeling also in programming DBS in general?
Absolutely. And I think it's really important to acknowledge how clumsy our metrics are.
Yeah.
And we try to be objective about it, and we have scales to try to quantify someone's motor function, but there's so much we're not capturing there, and even the best clinician is missing so much of even the observable function. But there's all this, you mentioned interoceptive, there are all the unobservable aspects of this, right?
Yeah.
That you how a person feels. Ultimately, at the end of the day, you want to improve people's quality of life. And I think there is a lot of the way that these kinds of therapies might be impacting people that are not going to be measured necessarily in always gait speed or hand opening, closing, velocity. There's more to it than that.
Yeah.
That resonates with me for sure.
Super cool. And then the other thought that I've, when talking to Julian Neumann, who and who also was on the podcast, he essentially said that, when the first poster was presented or abstract was presented in the meetings, as you mentioned, results maybe weren't huge, right? It wasn't like a huge improvement. But the other thing he said is that with FDA approval and this becoming more, now a thing, there could be so much more additional science to actually figure out exactly which parameters to set up and so on. Because so far, as I understand, it was quite exploratory, right? It wasn't yet clear exactly how to set all these settings and, which mode is better and so on. So I'm sure it's going to be a huge success for the field that now we can address these questions going forward, right?
Oh, yeah. And Julian's going to have such an impact on that. I mean, I'm really eager for him to get his hands on this and really start to explore it. I mean, I think we're going to learn so much. I mean, this trial, when you think back to when it began in 2020, and of course, the design began before that, there had never been somebody on an adaptive program for DBS that was outside of a immediately observed setting, right?
Yeah.
And they went all the way from often with externalized leads, right? It was the only data they had to go on, and they went all the way straight to having these things on device and people ambulating around, for ultimately years of follow-up. It was quite a leap. And no doubt we were, figuring out how to use these systems as we went. And so I'm confident that we haven't figured out how to get all the potential benefit out of even the current system, much less as you start thinking forward to what are the some technical advancements that could make this better in the future.
Yeah. Can you talk a bit about the complexity of the programming? Not so many people have had hands-on experience. I think the sensing capabilities are public and everybody can try those these days, but not the closed loop stuff yet. So how complicated is it? Will you go, will you offer it in the future to everybody or only to complicated patients, or any insights on that?
Yeah, other great questions. So I think a lot of the complexity is derived directly from the complexity of sensing. And so I think if you're a DBS programmer and you've been using sensing, and I think a lot of the early adopters are people that were already used to using this kind of data from the operating room or externalized leads, and so had already an intuition about neurophysiology data. But the neurophysiology data is complex. One of the limitations of the current tablet interface is that it's hard to fully assess the signal just from the tablet. So you can get a nice power spectral density, you can see a nice beta peak, but you're always looking at an average. It's hard to understand the time varying aspect of it. You're looking at the recording that's obtained, like the device can record in ways that are more resistant or less resistant to external noise. And what it shows you on tablet is always in the recording mode that's more resistant to external noise. So it gives you a better way to see the beta, but does not give you a great view into whether there might also be ECG artifact or other fluctuation, say in the noise floor that one of our for a synchronous component that you might, because the actual control signal, to be clear, is just a band power, just a band pass power. So anything else that encroaches on that can also influence your control algorithm. So I think the best way to assess that is really on tablet. Once you set your control signal, you can look at the power in that band as you adjust stimulation and demonstrate that if you expect that as you ramp up stimulation, you're going to suppress that. That's something you can very much confirm on tablet. But still, a lot of our programming also involved looking at data offline and looking at, for evidence of artifact and really understanding how stimulation affected frequencies outside of the narrow band that was being controlled and those kind of components. But I think that our experience in the trial, it's going to be easier in the real world than in the trial. Because in the trial, of course, you have a lot of constraints and there's a lot of things that you're supposed to test as part of the trial protocol. Plus, we were trying to set up both modes. So it meant we were often bouncing back and forth between the two modes and trying to set them both up because we were sort of exploring these modes. I think in real clinical practice, there's a lot of information that you can derive during your initial programming and as you're doing your initial titration of continuous DBS. So you'll already ideally know your control signal. You'll have seen how it responded as you titrated DBS. You'll have if you have in mind that you're going to program adaptive, you'll ideally have mapped out a little bit, have this sense about what your upper and lower stim limits are. In other words, your lower stim limit is probably defined by the lowest level of stimulation you ever want to deliver. So when they're in their peak on state, how low do you want it to go? Or since these signals tend to be low at night and you tend to be relatively low stimulation at night, consequently, how low do you want stimulation to be at night? And your upper stim limit is going to be either where you hit side effects or the most stimulation where at this point, when they're off medicine, anything above this doesn't seem to give any additional benefit. And so if you already have that in mind and you have your control signal and you actually come to set up adaptive, you've got a lot of the components already. And the other piece I think about the trial that's going to be easier in the real world is that I think that Medtronic learned a lot from the investigators about the interface.
Yeah.
So there are many things about the interface, for example, the way you adjusted these thresholds that were clumsy in the initial version.
Yeah.
And the version that has rolled out now in Europe and that we'll see here are just much easier to use and more streamlined.
That's great.
So,
that's great.
Like any new approach to programming, there's a learning curve with it. But I think if you're already using sensing information, I think it'll be digestible.
Yeah.
But I plan to. I'm obviously very interested in it, and I think it'll be really interesting to explore who this helps and who it doesn't help. So I'm eager to try it in any of my patients who are interested in trying it. But I particularly think that it will be more helpful to address, residual motor fluctuations in people that aren't adequately controlled in CDBS, or people that are experiencing side effects from CDBS that you can't adequately control with just adjusting the CDBS level.
Did you have one or two patient cases where that was very apparent, like just on a non-scientific, anecdotal, let's say, level, you would really see, oh, wow, this had
Totally. Yeah. I mean, I think a couple that one is actually a group of patients that I actually went into this thinking it was going to be challenging to program them, which are the people that are making frequent adjustments to their DBS settings. Mm-hmm. Now, at one level, you think that's the ideal person, right? If it's someone that feels overstimulated and turns it down, and they feel understimulated and they turn it back up, and they're doing that multiple times a day, that's like the ideal person, right? That you'd think should respond to this. On the other hand, I really worried that you wonder as a clinician, maybe it gets back to your point, like, is it our judgment or how often is the patient judgment really the thing that trumps? I said, well, how much of it is placebo? How much of it is like they don't feel well in that moment, and so making a change feels good, right? Even if the change is not necessarily productive if you were to do it in a blinded fashion. So I thought, well, they're not going to like going from having control to now their patient programmer just shows this little animated infinity sign and gives them doesn't even tell them what their stimulator is at, much less give them a chance to adjust it. And I had two people in the trial that were making those kinds of frequent day-to-day adjustments, and both sailed with ADBS and did well, and felt like they didn't need to make adjustments anymore. That's which great. For me was really telling. I thought, like, okay, that it definitely proved my bias wrong.
Yeah.
The other case that is maybe another kind of situation that is very familiar to people that program DBS is someone who had fascinating gait that seemed to at least in part emerge worse in the period after DBS, not immediately acutely, but in the months and years after. And where CDBS adjustments, if you went up further on stimulation, increase the current, you could worsen the fascination. But also if you backed off too far, you would worsen it. Okay. So there seemed to be a sweet point there where it wasn't just being caused by DBS, but where there was clearly this was a major residual problem. So this is someone who didn't do very well on dual threshold, or rather dual threshold felt similar to continuous DBS, but single threshold ADBS, their gait was markedly better.
Okay.
And in a way that I still haven't fully wrapped my head around why.
Yeah.
But enough that they went from being dropped off in front of the clinic always with the valet, so they had the shortest possible distance. Oh no, I'll just go park in the regular lot and walk over. I mean, really, it made a material difference. Wow. And that was, striking to me, and, maybe appreciate that this sort of stim level titration approach to figuring out if gait was being impacted by DBS was perhaps not adequate in this case. Uh, and also maybe hints at, one of the challenges with these side effects of DBS that emerge more gradually. And speech problems can be like this sometimes, and gait certainly, where in the office, in the moment, you turn up stimulation a little bit, their movements are faster, their tremors better, things look good, right? And then it's the next day or the next week that they say, my gait is a lot worse. That there may be a benefit for these slowly accumulating side effects. If you can jump up stimulation to that higher level, that gives you an acute benefit to other bradykinesia, rigidity, or short-term symptoms, but also drop down frequently so that perhaps some of those slowly accumulating side effects don't accumulate. That's
really
very
cool thought.
speculative.
Yeah.
And as I said, I haven't, to my satisfaction, really understood exactly how this is helping this individual patient yet. But it was striking enough that it made me also. I do feel like in the single and dual threshold debate, maybe this is going to become the new STN versus GPI debate. That the fact that dual was performed a little better and more people chose it is going to be like, well, dual's better, right? But there are people that in my experience clearly benefited from single threshold in a way that at least in my hands, I couldn't program them to get that benefit out of dual. But I'm glad that both are coming out.
That's great. That's great. I just had Rainer Birkenbach on the podcast, who's, you might not know, but is the newly minted CEO of BrainLab. Used to be the CTO, but since this year. And he mentioned from the early days, we talked about, when BrainLab was still a startup, essentially, their first customers were surgeons that were also engineers. They had, they were essentially partners in developing stuff. Now, of course, they really try to make it, an all-comers, tools that work where you don't have to be an engineer. Now, I'm drawing the parallel to you of being one of these, DBS programmers that have a background in even primate physiology and cell recordings, ePhys. You like this stuff. Now, how will it translate to the general neurologist, potentially, or at least DBS specialist that might not be into beta power and doesn't have the background of maybe understanding the whole literature and the bursting and all that? I guess one answer that many people come up with is AI is going to solve it. It will all be automated in the long term, but might not be that easy. And maybe you have some thoughts on how will Medtronic need, like, what has to happen now to make, because the push is to make DBS easier, not harder, right? Even for the caring clinicians. So any thoughts on that end?
Huge problem. I think on so many different axes, DBS is getting more complicated. So it absolutely needs to get easier. It's hugely underpenetrated. Even I would say that even the image-based programming, it adds complexity in terms of like how to interpret the imaging, and if you don't know the pipelines for how to put the imaging on the tablet and validate all these new tools that are potentially so powerful are just adding more complexity to this. And then sensing, compared to, you look back at the introduction of current steering electrodes, which I think similarly produced a sense of like, oh my gosh, it's getting even more complicated. And how am I going to do a monopolar review of now 16 contacts as opposed to it was already bad enough at 8? And, many people like Jens Volk been kind of developing like more efficient search strategies to use that number of contacts. I think this is going to be more challenging than that. The space is bigger. The variety across patients is going to be larger. That being said, I do think that what's going to happen, I predict, is that it's going to be these same centers that are all really, really invested in sensing, that are interested in trying to implement this because they either because they believe in it or because their curiosity drives them. And it will be a manual process. It's closed loop, but it's very much human in the loop, right? Like we're setting all the parameters manually, and I think that it can be efficient enough to be practical in a center that's invested in it. And I think increasingly, just in general, in DBS, it's having to be concentrated in centers that are really focused on it, because it's now, all the tablet interfaces are more complicated. If you're going to be a center that supports all the different manufacturers, if a patient walks in and has DBS, are you going to be open to any DBS system? It already requires kind of an investment in really understanding how this technology works. And I see that, like, that specialization as happening more over time, rather than this goes the other way, where this now needs to be taken over by a general neurologist. Any more than you wouldn't ask. General neurologist now is serving a role like a primary care physician in medicine, right? Neurology is getting so specialized that you wouldn't ask the primary care physician to program a pacemaker. Right. And so my view is that in the kind of short and medium term is going to need to continue to be concentrated. But my hope, now this is where it's going to be the hand wavy AI thing that you led with, right? But my hope is that ultimately it could be patients that are guiding this, right? Like that really will be more overseeing it and setting some parameters on it. But I hope we can reach a point where, in between image-based programming to kind of validate lead location and select what are likely optimal contacts, sensing, and maybe it's not just beta, but ERNA, other signals, identify, okay, these contacts are near the subcircuit we know is useful to suppress tremor or useful to suppress bradykinesia. And then algorithms that are just on device that explore a range of stimulation levels and look at these feedback signals. And when you come back, you're looking at a more and incorporate patient wearables. I mean, it's crazy to me how much data is already out there that we're not yet really incorporating into this process. That there's a vision of this that really is comes to you packaged. When you see the patient back, you have a whole nice display of all the data, and allows you to make kind of higher level decisions about the next steps, as opposed to having to tweak every threshold, every stim limit, those kinds of parameters. So I think it can get easier, but it's not on a 3-year timeline that's going to get easier. It's going to get harder first.
Sounds sensible. And the ECG artifact you mentioned briefly, there's, I think, one report, I think you're also on it, where that, the idea would be to implant on the right chest side for that reason. Is that happening? Just to be further away from the heart? Yeah,
Julian Newman led that effort and really gathered data sets. We contributed data sets to it. And that was really his. And he's actually developed really nice ECG artifact detection algorithms that he's open sourced. And we already were implanting on the right. The idea had been that in general, the pacemakers go on the other side, and so we kind of left that spot free. It wasn't driven really with this sensing in mind, but did have people on both sides and noticed that there's a clear difference in the artifact in the two sides. So I think if you're interested in sensing, implanting the DBS on the right makes sense.
Yeah, sounds good.
Yeah.
And then you also briefly mentioned different threshold or different beta activity at night or on the day. So there seems to be a circadian pattern there, which I think has only been discovered in the last maybe 1 or 2 years, if I am correct. That seems really important, right? Especially when setting thresholds. Can you elaborate a bit on that? Like, is that a real pitfall, or do people have it in mind, or could you even think of adapting the thresholds over the activity, or how is that currently been dealt with?
So I think this is a really fascinating underexplored area. A lot of people are doing research now trying to understand how DBS impacts sleep. How should DBS be optimized for sleep? But I think you really don't know. Within the current constraints, unless someone were to, when they go to bed, switch to a different group, there is no way of the system automatically detecting sleep and switching to a different program.
Yeah.
Right. So you really are applying one set of parameters to the 24-hour day. What that means in practice, since most patients have significantly less alpha beta power at night compared to during the day, is that that's usually going to be a downward driving force on your current, so that you will, in single threshold mode, have a lower duty cycle, and a dual threshold be closer to your lower stim limit. And so I think that that could be a real problem if you found that people needed sort of maximal stimulation when they're sleeping.
Yeah.
Actually, if anything, people tended to sleep more, in my experience, with adaptive, and I actually think that probably there's room to back off on stimulation at night, particularly if when people wake up, the system can adapt quickly enough to return to whatever their more optimal waking setting is. So I think you, practically speaking, need to set your lower stim limit with a consideration of what's the lowest level of stim you ever want to give during the day and night, and you have to choose whichever one is the higher.
Sounds good.
If that makes sense.
Yeah, we covered lots of technical hurdles already, and also lots of the outcomes of the trial. Just to be like complete, are there any other points you wanted to make in either, outcomes from the trial that we haven't covered, or other issues like the investigators had?
Yeah, let's see. We covered a lot. I think the other one that always comes up, posters and talks and whatnot, is the TEED question, right? Overall, how did the adaptive approach affect the amount of energy usage of the system? And overall, both modes had slight reductions in TEED, or total electrical energy delivered.
Yeah.
So, in general, people were receiving adaptive, and they overall got, on average, a bit less stimulation than what they were getting on CDBS. But the impact on battery life is more complicated, because there's obviously a cost to having sensing on all the time compared to not having sensing on, and there's a cost of the adaptive algorithms operating. So that is going to end up being individual. With some patients on adaptive compared to CDBS without sensing, we'll see improved battery life, and some people will see shortened battery life, which are rechargeables, might not matter so much. The primary cells, like most things, it's going to require a conversation between the clinician and the patient about whether if they do experience shorter battery life, whether the potential upside is worth it for them. Yeah. But I think that's something also we'll learn a lot more about in real world practice.
Makes sense. Great. And then maybe last question on this, where do you see the more general adaptive DBS headed, also maybe above and beyond Parkinson's disease, for other disorders?
Yeah, that's, I think, there's so much fascinating work. I mean, this is going to, in the same way that ambulatory sensing enabled DBS is already fueling a lot of discoveries, I think having these algorithms out there is going to let people test a lot of new ideas. I'm really excited about the psychiatric space.
Yeah.
Right. So I think Samir Sheth and Nicole Provenza and Mark Richardson and Darren Doherty and they've kind of converged on some of these somewhat lower frequency signals, alpha band signals in OCD that seem really fascinating and are still a little bit mysterious, right? Helen Mayberg, sort of broader, like longer term signals affecting healthy versus dysfunctional states in major depressive disorder. Fascinating by that. Again, very different scales, right, of which these are operating. Both of these are biomarkers that were more evident not as necessarily moment-to-moment biomarkers where you titrate stimulation to address it in that, the next second, but rather biomarkers where patterns across longer periods of time really are what reflected response.
Yeah.
And I think that's, to me, a harder to explore part of the sensing landscape, and potentially hugely important. Like, it's easier in the moment to say, like, well, what signal correlates with your hand opening and closing speed? But they're potentially much more subtle signals present in the data that correlate across these longer term trends. So I think exploring those, understanding how we can develop adaptive algorithms that can track those kinds of signals over time and maybe adjust on a totally different time scale, I think that's going to be really exciting.
Tourette's as well. Fascinating stuff right out of Florida. I think had Isaac Gundus on the talk series recently, but also on the podcast before. I think that's maybe also another very promising area. But I agree with you. And then as you say, it's like these new, like just having the device out in the field will empower a lot, right? It's always been the case for the field of DBS that new technical possibilities will lead to lots of discoveries, and this is going to be a big one, I'm pretty sure. So I want to be mindful of your time and maybe conclude with some rapid fire questions. Feel free to answer short or long as you want. How does the future of DBS now look in 10 to 20 years?
Yeah, I'm glad you said 20, because I think things don't change that much on a 5 to 10 year time scale. But I think my hope is that in 20 years, DBS is a lot more complicated than it is now, but a lot simpler for the clinician, right? That we've been able to figure out enough and develop these algorithms and supports enough that the systems are able to interact on to treat all these different symptom subdomains. Can they integrate your tremor responsive network and your bradykinesia responsive network and physiology that corresponds to each of these, and deliver that to you as a clinician in a well-packaged way that incorporates all that information. And hopefully we've unlocked some of these other approaches to stimulation that work fundamentally differently, right? Similar to these optogenetic manipulations that really offer much longer lasting effects, but with less risk of ongoing stimulation related side effects. That'd be my hope anyway.
Sounds great. Any eureka moments or also unexpected scientific insights that changed your thinking?
I know you always ask the eureka question. I don't think I have. I don't have my my fungus in the dish, mold in the dish, like penicillin moment, right? Many eureka moments with patients. Not yet, exactly, but nothing that I would highlight is that kind of level of eureka.
Got it. I mean, any time, like maybe the opposite is also good to cover off, like things that were a waste of time or didn't go as well. So,
yeah, of course, there are many of those things, right? Every project that you start, every analysis you do that doesn't work, I guess I don't tend to view them as wastes of time. I sort of I view them all as just part of the journey. And even if you start a big project, you spend a lot of time, it doesn't go anywhere, that you learn something along the way. And maybe I'm not quite end product focused enough, so it didn't seem like a failure. It felt like the journey was the success.
Almost everybody answers that way, right? That there's no waste of time, and I still kind of keep asking it that way. Maybe I should just change it, but I also I find it interesting to reiterate on that most people in academia are used to these things where sometimes you spend time on something and it doesn't lead to direct output, but normally you learn something, and that's, I think, good to hear for also the younger listeners. That's maybe one reason but almost everybody says that. So that resonates with me too. What advice would you give to younger folks that want to enter either DBS, neuromodulation, science, academia, or also neurology at large?
Yeah, I do like meeting with younger scientists, people that are thinking about these career trajectories and advising them. But I'm loath to give general advice. I
Sure.
don't feel like I come from a position where I have a singular piece of advice that applies to everybody. I guess I'm more Socratic, and I like to ask a lot of questions and try to figure out what their particular needs are. But I guess I would say this is an incredible time. If you're listening to this podcast because you're maybe thinking about entering this space, this is an incredible time to enter neuromodulation, where it from an academic perspective, from a clinical perspective, from an industry perspective, there's so much happening right now, and it's clearly going to be continue to be an explosive growth area.
Yeah.
I would say if you want to do this kind of work, full steam ahead. It's a great time to dive on in.
Sounds great. And then what would be biggest missed opportunities in our field? Things we should be doing but are not enough?
Yeah. For me, the one that springs to mind is just the amount of data that's being collected that is impossible to leverage right now. All these sensing enabled devices that are recording data that right now just sits on our servers here and everywhere else, and all of I was thinking about, like, if we could unlock in some way all the data that's just flowing around us, like every patient that has an iPhone and probably an Android phone. I'm just not as familiar with the behind the scenes, but has a longitudinal record of their gait speed going back years. And if we get access to that data and look at their DBS settings over time, like, there's an amazing study right there, just looking at how gait changes over time, people with Parkinson's, how DBS parameters affected that. But so you have that idea, but then, like, getting that data and getting the kind of privacy part through the IRB, and just by the time you only have so many hours in your day, right, to get a hold of that. So some way to unlock this potential, start putting together bigger structures. Now, some of it I think is really a lot of interest. Watch Rune Labs and their Strive Study and Strive PD platform, right? It's a drive towards that, right? Let democratize this, let patients contribute their data. I think we need to have better ways for them to contribute their DBS data, right? But with the sensing enabled data, if we can kind of figure out a way that we can merge that, and I think maybe address some of the privacy concerns by putting it with the patient to let them share their data, and maybe we could start to really kind of put some of this together. But it's also tricky from an analytics standpoint, because real world data is noisy. And so you always have this debate: are you better off collecting carefully controlled data and less of it, or a lot of real world data? And there's people that are like, well, the real world data is more reflective of real world behavior, and so it's more valuable. And there's people that are of the garbage in, garbage out mentality, right? And it's just so noisy that you can't understand it. I tend to be more optimistic that data is going to be really important, and we can capture big trends there. It's just so hard to get it.
And maybe if you really want to change things, you have to kind of show it with real world data, right? There's a danger if it's meticulously controlled lab data, as maybe the first closed loop, you mentioned Simon Little and Peter Brown studies they were very important, but they would probably not automatically generalize to the bigger public domain. So very much agree. And also, that's a big challenge, but I also liked your idea of the Rune Lab idea of, like, essentially the who owns the data is really the patients, and then driving awareness of maybe them sharing data or making them available for research would be super helpful. So thank you, Todd, so much for all your wisdom and sharing that with us. I as expected, it was very enlightening. Even though we've been colleagues for a long time and been following your work, it's always great to sit down and really take the time to talk. I learned a lot. Is there any topic you wished we had discussed but we didn't?
No, this is wonderful. I really appreciate the chance to have this conversation with you. And maybe the only thing I'll add is I'm so appreciative of your contributions to this field. And I'll say that you taught me DBS anatomy. The lead DBS
wow.
platform, which I started, early on in residency playing with, and then, that is shaped, and I think I'm not alone there. Shaped so many people's understanding of how this therapy works. I have those models in my head all the time, spinning around in my head when I'm thinking about this, and I think you have driven that revolution.
Wow,
And
that
so, thanks
means
for
a
all
lot.
that, and yeah.
I really try to make the podcast not about me. It's I'm of course very honored for you saying that, especially somebody like you, that I so much respect as a scientist saying this. So thank you. And maybe as an anecdote to still share is that I think you were one of the, or maybe the first contributor of code from that was not in Berlin to lead DBS. So you did actually back in the day contribute code, and there's still parts of it in it that were written by you yourself. So thank you also for being an early adopter of the DBS. That's really, really great. Yeah.
To my great benefit. Thanks, Andy.
Thanks so much. All right. Great.